Maryland Life Sciences would like to recognize today's supporting sponsor, Johnson & Johnson Innovation. J Labs at Washington, D.C. is a global life science and healthcare incubator network that provides early-stage innovators with access to all their needs. Located on the new Children's National Research and Innovation Campus, J Labs at Washington, D.C. also serves as the hub for Blue Night, a joint initiative between J Labs and the Biomedical Advanced Research and Development Authority. Our contributing sponsor, GSK. At GSK, we unite science, technology, and talent to get ahead of disease together. We prevent and treat disease with vaccines, specialty, and general medicines. For more information, go to gsk.com. Our keynote sponsor, DPR Construction. As the #1 pharmaceutical builder in the country for the last 5 years, DPR constructs cutting-edge spaces that challenge norms and foster discovery, allowing our heroes in lab coats and Tyvek suits to change the world. We bring integrity, enjoyment, uniqueness, and an ever-forward mindset to each engagement. DPR is ready to partner in building great things, great teams, great relationships, and great projects. We'd like to thank these and all of our sponsors for their continued support of the Maryland Life Sciences Bioinnovation Conference. Our next panel will discuss factors impacting patient access to cell and gene therapies. Please welcome your moderator, Dr. Cartier Esham.
Good morning. How is everybody today? I know I stand between you and lunch, but I promise this will be a very exciting discussion. If I could just ask everybody to quiet down just for a minute. Thank you. My name is Carter Esham. I'm the Chief Scientific Officer of the Biotechnology Innovation Organization, or BIO. I'd like to thank the Maryland Tech Council and those involved with organizing this important conference for hosting this very important discussion on factors impacting patient access to cell and gene therapies. I'm going to take a few minutes of opening comments, and then I'll ask each of my two fellow panelists, Nina Hunter and Colin Werth, to introduce themselves and provide their opening comments, and then we're going to have a discussion. The first thing I think that's important to raise to sort of set the table for this conversation is the fact that it's a very exciting time for cell and gene therapy development and the amazing potential they have to improve the lives of patients and their families. The fact that we're able to use the term cures in and of itself is truly extraordinary. Presently, there are over 2,400 cell and gene therapy clinical trials in the pipeline. And to put this in context, in 2020, BIO calculated that for gene therapies alone, there were 230 development programs in the pipeline, compared to 93 in 2015. This is a fast-growing area of science. Most of these clinical trials are in phase 1 and phase 2, so the other thing that we're going to be seeing is the establishment of new precedents, the establishment of new understandings that are going to be foundational for charting the path forward for these medicines and getting them in the hands of patients who need them, which in many cases means their first available treatment. So with that background, in late 2020, Bio, Pharma, and the FDA engaged in the 7th technical negotiations for the Prescription Drug User Fees Commitment Letter, or PDUFA 7. This occurs every 5 years, where biopharma and FDA work to determine how to set and provide resources to ensure the effective and efficient review of products at the FDA. In fact, the PDUFA 7 agreement was just enacted by Congress last week. This is the most CBER-focused user fee agreement in history. While PDUFA 7 will provide many improvements for all medicines, such as improving processes to enable scientific dialogue throughout the entire development and review processes, there's also going to be improved engagement specifically relating to CMC readiness and review and enabling the utilization of innovative manufacturing technologies, which of course are very important to cell and gene therapies. CBER will specifically be able to bring on substantial increases in staff, capacity, and capabilities. PDUFA 7 will also advance patient-focused drug development, including advancing the utilization of patient preference information and use of patient experience data for benefit risk assessments and product labeling. CBER will hold a dedicated public meeting focusing on the current and future needs of capturing patient experience data and patient preference data as it relates specifically to cell and gene therapies. This will be important in informing all future guidances relating to advancing patient-focused drug development. There are pilot programs that will improve our ability to use real-world evidence to advance understandings about what is needed to support efficacy endpoint development for medicines that treat rare diseases. And lastly, PDUFA 7 provides significant resources to expand capacity needed to advance the use of digital health technologies, modernize its IT and cloud capabilities, and there is a particular commitment of significant resources for CBER to improve its bioinformatics and computational capacity, which is critical for their ability to support review and submissions for next-generation cell and gene therapies. The last point I'd like to address, again to set the table for this conversation, is the fact that these transformative therapies may be individualized applications and require unique clinical circumstances which challenge today's payment systems. Patients are and will continue to be treated across varying healthcare systems and settings with differing associated care needs by specialized providers. As we work to bring patient access to innovative therapies, more work must be done to modernize the healthcare system to meet this goal. The new wave of transformative therapies are providing significant, durable value, encompassing patient healthcare outcomes, delivery of care, and overall healthcare spending. These medicines have the potential to truly transform the treatment of serious diseases, rare diseases, and other life-threatening conditions. But patient access will remain uncertain unless we address the following issues. First, we just have to modernize the reimbursement system. Outdated coverage, outdated coding, outdated reimbursement pathways, particularly in Medicare and Medicaid, must catch up to the fast pace of innovation and ensure patient access once a therapy is approved by the FDA. But when we say we must provide access, we must ensure that there's timely coverage and access. Timely coverage is a top concern both in Medicare and Medicaid. As an example, current law requires that Medicaid cover drugs that are medically necessary and for which there's a manufacturing drug debate agreement in place. However, despite this clear mandate, many states impose arbitrary procedural delays, particularly for those, we see this very often in medicines associated with FDA's accelerated approval pathway. All patients deserve timely access to FDA-approved medicines. Third, we must enable paying for value. Much of our healthcare system operates on a legacy construct where payments were based on volume, not value. But because of many of our transformative therapies today and in the pipeline are administered once, where their effects are expected to last a lifetime, there's no way to tie a payment at the time of administration to measures of ongoing effectiveness as we would have under a value paradigm. So we need to address and remove barriers to value-based arrangements, where payments are tied to the value the therapy provides to the patients and to the healthcare system. BIO is committed to working with patients, policymakers, and other stakeholders to ensure successful implementation of PDUFA 7 and to developing and advancing innovative payment models and financing approaches to ensure timely patient access to these next-generational, game-changing medicines. So with that, I'm going to turn it over to my colleague, Nina Hunter, Vice President of Corporate Strategy for REGENXBIO. Nina, I'll turn over to you, and then we'll go to Colin after you complete your opening comments.
Thank you, Carter. Do you have my slides there? Will that be pulled up?
I see them.
Okay. So, do I just say next slide? Sorry.
There you
okay.
go. Yes, there you go. You're ready.
Okay. Thank you. Hello, my name is Nina Hunter, and I'm VP of Corporate Strategy at REGENXBIO. As Carter said, collaboration is key to developing therapeutic options for patients, especially in rare diseases. So today I'm actually speaking on behalf of the Pathway Development Consortium, or PDC. The PDC is a nonprofit public-private collaboration co-founded by REGENXBIO and Solid Biosciences last year. And I will share some of the scientific and policy work we've been doing since the consortium was launched, specifically to support AAV gene therapy development. Next slide. The PDC was established to guide the recent decades of AAV gene therapy research into a future of innovative, potentially life-saving therapies. The overarching PDC goal is to foster collaboration and partnership among patients, industry, regulators, academia, payers, and other stakeholders. Today, we have a vision to construct ideal pathways to ensure that all born with serious genetic conditions can find their way to effective AAV gene therapies. We have been actively pursuing topics to accelerate the development of AAV gene therapies and are currently working to establish more formal governance to expand the collaboration of the PDC to include other members. We are mindful to stand up an inclusive nonprofit that speeds things along and to involve as many stakeholders as possible so work is reflective of the wealth of experience available. We recognize there are other public-private collaborations and partnerships in existence with goals that complement the PDC's mission, and we strive to ensure that the PDC's work does not duplicate existing efforts. Next slide. So one of the focus of our consortium has been accelerated approval, an important regulatory tool for expediting the availability of these products to patients. One of our initial activities was to develop a proposed framework that could be applied to AAV gene therapies to facilitate the use of the accelerated approval pathway. Our goal is to provide a systematic way to consider different types of indications and the general type of evidence needed to support the use of this pathway. A key determination for use of this pathway is the identification of a surrogate endpoint reasonably likely to predict clinical benefit. In the case of AAV gene therapy, many diseases being pursued and have well understood disease pathophysiology, and mechanistic evidence can be an important contributor to the determination that an effect on a biomarker can be used as a surrogate endpoint. A stepwise approach can be used to support the determination that a biomarker is reasonably likely to predict clinical benefit. First, demonstrate that the presence of the measurable biomarker, for example, a protein. This could be clinical data demonstrating that the protein is present in the intended cell or tissue after administration and can be reliably measured. Next, provide evidence that a change in the biomarker would lead to a clinical benefit. One can leverage data known about AAV gene therapy to model how AAV gene therapy administration can achieve the needed protein levels that represent a change that is reasonably likely to provide clinical benefit. The key is then linking these steps by relying on clinical data, and when not available, relying on preclinical data, including in vitro and animal data, to model the correlation in humans. We released this framework as a draft and are hoping that the community will review and provide feedback to make it more useful. It is posted on our website, and let me just walk you through some of the details and examples in the next 3 slides quickly. Next slide. In the proposed framework, we thought about rare diseases with underlying genetic defects and categorized them generally into 4 groups. These 4 groups run across the top here. So for example, monogenic diseases leading to defective or missing protein, and that missing function is either cellular non-enzymatic, cellular enzymatic, non-cellular non-enzymatic, or non-cellular enzymatic. Of course, this is just one way to categorize the disease and not the only way. And the 4 categories can be thought of as a 2x2 table based on whether the protein, for example, is affected by genetic defect and whether the targeted by gene therapy is inside or outside of the cell. If the protein is outside the cell, its level could potentially be measured in circulation, whereas if the protein is inside the cell, tissue biopsy would likely be needed to evaluate its level. Next slide. So here are some examples of how this can be applied to actual diseases and conditions. So for Duchenne muscular dystrophy, this is an example of a cellular non-enzymatic disease. DMD gene mutations result in alteration of the structure or function of dystrophin or prevention of any functional dystrophin from being produced. The lack of dystrophin within repeat muscle contractions and relaxation leads to damaged fibers that weaken and die over time, resulting in progressive muscle weakness. Dystrophin plays an important role with the dystrophin complex strengthening the plasma membrane of striated muscle cells, And dystrophin is a protein that links intracellular cytoskeleton network to the transmembrane components of the dystrophin-glycan protein complex. AAV gene therapies seek to restore function by delivering microdystrophin to act as dystrophin. So in this category, we can measure the actual protein that is being restored as a surrogate endpoint, microdystrophin, through a muscle biopsy in the tissue. Another example in general is lysosomal storage diseases, which are good representatives of the cellular enzymatic category. Next slide. On this slide are just two examples to represent the non-cellular, non-enzymatic, such as hemophilia, and non-cellular enzymatic, such as maple syrup urine disease. I won't go through the details here, but again, please take a look at our proposed framework and provide any feedback. Next slide. And I'm sure some of you are familiar with what's shown here. These are published recommendations from FDA on consideration to support the use of a surrogate endpoint. These are not all of the recommendations. I just pulled some here as examples for reference. Next slide. Now, besides a framework, the PDC has also been focusing on a different work stream, on Duchenne muscular dystrophy specifically. After establishing the consortium, we kicked off with a roundtable on defining clinically meaningful clinical trial endpoints for the range of functional statuses of the Duchenne patient. Our overall aim was to promote scientific and policy interchanges among a broad range of Duchenne stakeholders. In building on the discussions at that roundtable, we issued a white paper which identifies areas where attention is needed to facilitate development of AAV gene therapies for Duchenne. Our plan again was to work collaboratively to address these issues. And both the meeting summary and this white paper are available on the website. And most recently, in December of 2021, we launched a working group to implement some of the priorities articulated in the white paper. We're expanding efforts by further exploring and outlining an approach that leverages the accelerated approval pathway for development and approval AAV gene therapy products as therapeutic options for patients with Duchenne. Next slide. For this work stream, our Duchenne use case allows us to explore the framework more closely. We identify 5 topics to start: the appropriateness of accelerated approval pathway in Duchenne. It's a serious disease, and how therapy will be able to provide meaningful advantages over other therapies. The criteria related to accelerated approval, demonstrating effects on an endpoint reasonably likely predict clinical benefit. And here we had 2 subgroups, each working on justification, the use of microdystrophin and fat fraction using MRI-MRS techniques as promising surrogate endpoints with preclinical, natural history, and clinical data to support their use. Other endpoints that can be supportive in making an efficacy determination, so considering and identifying those that are most useful, potentially data from real-world settings, such as video assessments or wearables, and other clinical outcome assessments. And then lastly, one important key to use of accelerated approval pathway is the timely completion of studies to verify and describe the anticipated clinical benefit and benefit risk considerations. Next slide. So as noted earlier, the key to success of use of the accelerated approval pathway is to verify and describe clinical benefit, and regulatory flexibility is important to address unmet medical need. But ultimately, what is important is a demonstration of clinical benefits that are meaningful to patients. Next slide. I won't go through these here, but these are some points about post-approval studies and what they're intended to verify and how they're important. And lastly, with that, we, the PDC, look forward to additional work groups in the future as we continue to work with our partners to provide a robust forum for these types of scientific and policy exchanges. Thank you for your attention.
Thank you, Nina. That was really informative. Thank you so much. I'm now going to turn over to our fellow panelist, Colin Werth, who's a patient advocate, who's going to remind us why this matters and help us better understand what it means to be advancing patient-focused drug development. So, Colin?
Thank you for the introduction, Cartier. Glad to be here today, and thank you for the Maryland Biosciences Consortium for allowing me to speak today. So my name is Colin Werth. I'm a lifelong resident of Farmville, Virginia, located just over an hour west of Richmond. I have an associate's degree in information technology from Southside Virginia Community College. I've been working for the past 7 years as an IT specialist for Letterpress Communications, a marketing firm based in Farmville. In addition to my work in the IT and marketing field, I'm an advocate for the rare disease community. I was diagnosed with Duchenne muscular dystrophy at age 3 in 1998. So Nina actually gave a great overview of Duchenne. As she mentioned, Duchenne is caused by mutations in the dystrophin gene, which is actually the largest gene in the human genome. So, I can talk more about the progression of Duchenne. So it is a progressive disease that gets worse over time and is in the end fatal. Typically, you first lose the ability to walk, then to feed yourself and to breathe, and eventually develop heart failure. But thanks to many years of research and improved care guidelines, the life expectancy for those living with Duchenne is increasing, with many living into their late 40s and early 50s. About 1 out of every 5,000 boys born has Duchenne, and about 20,000 worldwide are born with it each year. Duchenne mostly affects males and reaches across all races and cultures. It is estimated that there are about 15,000 boys and men, as well as a few women living with Duchenne today in the United States. And that's my introduction.
Thank you, Colin. I just wanted to make sure
that our conversation.
Thank you, Colin. That was lovely and informative. Why don't we start the conversation with you? So again, you talk about your experience as a patient advocate, but how has access to a patient advocacy group, benefited you, benefited your family? What would you tell the audience about patient advocacy groups?
So my parents connected with the patient advocacy organization, Parent Project Muscular Dystrophy, or PPMD, soon after my diagnosis of Duchenne. PPMD was founded in 1994 by Pat Furlong, mother and nurse who lost 2 of her sons to Duchenne. Thanks to the support of PPMD, I've been able to receive care from some of the best doctors in the country. I personally became involved with PPMD in 2016 by joining their Adult Advisory Committee to help provide guidance on Duchenne-related programs as an individual living with the disease.
And I can personally say that Parent Project Muscular Dystrophy has been one of the leaders, really leading groups in advancing specificity around patient-focused drug development, including patient preference data. So it's a truly important voice.
Yes, definitely. And so I'm a passionate advocate for Duchenne policy and research efforts. I've worked with multiple pharmaceutical companies, both in the U.S. and internationally, to integrate the perspective of the adult community in their drug development and access programs. I'm currently serving on a clinical trial advisory board of a major pharmaceutical company. PPMD has been a vital resource for the Duchenne community and an upstanding advocate for research and policy related to Duchenne. Passage of the 2001 MD Care Act by Congress would not have been possible if it weren't for the efforts of PPMD. In addition, PPMD has been instrumental in bringing treatments to patients like me by gaining over $600 million in federal funding for research over the past 20 years. Thanks to these efforts, there are currently 5 FDA-approved treatments for Duchenne, 2 of which I'm currently receiving. This is an amazing accomplishment considering when I was diagnosed nearly 25 years ago, there were no treatments available. There's still a long way to go before a cure is found. Gene therapy for Duchenne is in early stages of trials in patients. The current FDA-approved treatments help to slow or stop the progression of Duchenne, but with gene therapy, there is the hope of restoring lost function.
Well, and what is the one piece of advice, Colin, that you would give to companies developing gene therapies?
I would tell them to, make sure to include the entire patient population living with a certain rare disease, such as Duchenne. Often clinical trials for diseases, rare diseases with Duchenne, only include younger patients who are still ambulatory. This is because many popular tests to show drug efficacy are related to ambulation, such as climbing stairs, running 10 meters, or the 6-minute walk test. However, this leaves out the patients like me who are further along in disease progression and are unable to walk anymore. It's important to make sure that we are not forgotten. As patients living with progressive diseases, we are willing to take any treatment we can get to help us.
And Nina, sorry, Colin, go ahead. I didn't mean to interrupt.
Not being able to walk is one thing, but when you lose your arm function, practically all your independence is taken away. So just make sure when you're planning trials to make sure no one is left out.
Very sound advice. And Nina, from your perspective, you talk a lot about collaboration between sponsors and patient groups. So, what's your viewpoint on what that collaboration should look like?
Yeah, so with our work in the consortium, we have been working with PPMD as well and others to make sure that we're capturing that wide spectrum of perspectives in terms of how a patient might think about benefit risk. That's a decision that is very different for each patient and their families, as well as what's clinically meaningful to them, as Colin spoke about. Benefit, right? And depending on the stage of the disease and what's most important with day to day, that's where I think the video assessments and wearables, for example, might be helpful in supporting some of those endpoints as they're being developed for future use.
So in your presentation, you talked about how many of the cell and gene therapies, working on first-time treatments for rare diseases, this often means, and I alluded to it in my opening comments as well, often means that they use the accelerated approval pathway. Would you like to comment on the value of that pathway and what it means in terms of benefit risk considerations and what it means in thinking about long-term safety and efficacy issues?
Yeah, so I think accelerated approval pathway is an important tool, and FDA has many tools to help support the development of any disease, right? But when it comes to rare diseases and diseases of high unmet need, it becomes especially important because it means that the patient may have access to the drug sooner. And so FDA grants approvals to drugs and biologics that have demonstrated substantial evidence of effectiveness. And that can take a long time depending on the disease course and what's being measured. And so for use with the accelerated approval pathway, you can rely on a surrogate endpoint that is reasonably likely to predict that clinical benefit and use the post-market approval studies as well as long-term follow-up, specifically in the case of gene therapy, where long-term follow-up is required. So I think it's in a unique position to be able to leverage both the long-term follow-up studies for safety and efficacy to confirm that clinical benefit.
So and I'll put this up for both you, Nina and Colin, in the PDUFA 7 agreement, there are specific activities that, once you have that agreement in place, it leads to very specific actions, public hearings, the ability to comment on guidance, workshops, et cetera. And in this user fee agreement, there's a couple of things that seem relevant to me, and I'd love to get your all's thoughts on their importance or how you might be thinking about working with the FDA over the next 5 years on a couple of these provisions. One is the Rare Disease Endpoint Advancement Pilot Program, and that is designed to provide a number of applications each year, sort of additional more intense engagement with FDA reviewers and senior staff. And it's designed to sort of have those specific examples enable broader learnings to the entire industry, where those learnings sort of are anonymized somewhat, but again, it's sort of to give a broader understanding of what is necessary to bring forward an understanding of what is needed to support a novel endpoint for the basis of approval for rare diseases. There's also sort of tied to that, though, something that was put in place in PDUFA 6 that maybe is not often talked about, and I think we're still evaluating how effective it is, and that is that there's the ability of sponsors to have a dedicated meeting with reviewers and senior FDA staff to talk about novel surrogate endpoints and the plans for utilization of a novel endpoint and the plans for what's going to be brought to bear to support that utilization of that endpoint. So in the collaboration, Nina, that you're working on, how does that, do you view that as are you going to be sharing your learnings with those learnings? How do you view that sort of collaboration over the next 5 years? And then I'd like to hear from Colin, knowing that Parent Project Muscular Dystrophy is also, again, a very important voice in sort of advancing our understandings about these issues. I'd like to hear from him as well. So, Nina?
Yeah, no, so I think there are a lot of provisions that are coming that will hopefully move the field forward. As you mentioned, endpoints are an important part of the discussion, but so are other things such as flexible trial design and number of trials needed and statistical considerations when it comes to rare diseases and what's strong enough natural history to be able to support the data that you have. So I think having that focus around rare disease development will be very important and will help them cell and gene therapy field move forward. But specifically around endpoints, I think there are two different paths. So there is to clinically validate an endpoint, which you would use in a traditional approval pathway, so how patient feels or functions. And then there's the use of a surrogate endpoint where it's not validated, right? And so what is that reasonably likely? What's enough to pass that bar in order to access that drug sooner through the accelerated approval pathway, as you then wait out and confirm that benefit longer term through the confirmatory trial. So I think having discussions with FDA about both pathways and being able to pursue both would be great for rare diseases. But I think right now, focusing on the use of accelerated approval pathway on surrogate endpoints is one focus of the consortium. And we definitely plan on sharing all of these learnings through our website and through public workshops that we plan to hold in the future.
And Colin, I know you guys do a lot of thinking on just generally as a patient, what are risk-benefit considerations and what should they be in terms of clinical trials? Would you like to expand on that?
Yeah, so as any patient living with a degenerative medical condition which currently has no cure will tell you, having any option to improve quality of life or delay onset of symptoms is a game changer. With that being said, patients living with rare diseases definitely have more of a risk tolerance than the average person. So yeah, I mean, like, what is there to lose when living with a condition that already causes so many problems? Life is terminal for all of us, but why not make the most of it, enjoy as much of it as you can? That's not to say we should throw caution to the wind. There are so many unknowns with gene therapy being in early stages, so that's why we need continued state and federal funding, especially for medical research, to understand more how these treatments work and eventually bring access to patients.
So both of you in those comments sort of mentioned, those comments and previous comments, the importance of really thinking about clinical trial design and execution. One thing, there's been a lot of conversation since during the pandemic, sort of out of necessity, there was a whole host of approaches that had to be deployed, again, out of necessity. And it sort of leapfrogged, I think, understandings of how to work with regulators to utilize things like decentralized trial approaches, to utilize remote monitoring, to utilize digital technologies, to really think about how you design a trial and what that means in terms of a patient engaging in that trial. We've been trying to work with patient groups to capture learnings about what from the patient perspective did they view positively in those circumstances and where are there areas of improvement. So in terms of barriers for clinical trials, Colin, what are your thoughts on maybe some of these new approaches? What are barriers that are of most import to you and your community? And then Nina, I'd like to hear your response as well. Colin?
So yeah, one thing I would really like to point out is to pharmaceutical companies that realize that traveling for medical care and to participate in trials is especially difficult for someone with complex needs like me, who needs, and I also use a wheelchair full time. So, and anyone knows how complicated travel logistics can be for anyone nowadays. So add on top of that a complex medical condition, travel planning definitely becomes more daunting. So if you can, try to eliminate as much travel as possible, or even consider having patients with extended stays in apartments, such as near trial locations, so patients don't have to travel back and forth from home. Because when I travel, I have a lot of equipment. I have a machine I use to breathe at night, so just a lot of extra things to worry about there. So just consider that.
Nina, any comments?
Yeah, so I think the FDA has always been open to incorporate the patient perspective in regular decision making, and in the past, lots of programs have been focused on this. And I think, as you said, Carter, the learnings that we've had over the past 2 years around how to collect data from patients and how to use that information for regulatory decision making, is we have an opportunity now to really think about how to lower the burden for the patient, as well as be able to measure the day-to-day, which actually might be more accurate than some of the assessments that you might get when you go into the clinic for those studies, right? So perhaps using apps or like I mentioned before, wearables and video assessments to be able to capture that information automatically, that's probably even less biased, and how to use that evidence to support decision making, I think is really important.
Thank you.
And I'll say,
go ahead, Colin.
Sorry. And to add to that, yeah, definitely having to travel to different clinical site, that can definitely have effects on results as well, since like with traveling, it just can take a lot out of you, so you might not be getting the most accurate results. But if you're doing day-to-day, that definitely and that might be more beneficial.
Mm-hmm. Excellent points. And Nina, I recall when we were preparing for this conversation, I believe you had some interesting insights on some other collaborations, like the Bespoke Gene Therapy Consortium, as well as another program at FDA, the ARC program. And I wanted to make sure I gave you an opportunity to speak to this audience today.
Yeah, so the Bespoke Gene Therapy Consortium is something that was also launched last year, and many of you probably know about this. It's actually a public-private partnership, including NIH, FDA, and lots of pharmaceutical and life science companies, as well as nonprofit and patient organizations. And this is being led by the Foundation for the NIH, and the goal of the consortium is to improve current models for developing therapies around AAV gene therapies. And so one of the goals is to improve the understanding of the basic biology of AAV gene therapy, as well as the manufacturing and production quality control testing to help promote AAV gene therapy development. I should also mention that this is for specific diseases that have no commercial sort of pipeline being pursued, right? So these are ultra rare diseases that have no existing gene therapy under development, but the consortium wants to be able to pick some pilots from some of the applications that went out to be able to support the development of AAV gene therapy for these ultra rare diseases. And through that, share the learnings broadly with the AAV gene therapy community so that others can leverage some of those learnings, perhaps in a platform approach manner, to be able to then want to develop gene therapies for other ultra-rare diseases. And it's a 5-year project, and was launched last year. And in the coming months, the diseases that will be chosen for the pilot program will be announced.
Thank you. So we've got about 5 minutes left. I wanted to make sure that I offered an opportunity if there's any, we probably get 1 or 2 questions from the audience, and then I want to make sure to reserve sort of a 30-second closing thought from my panelists. But if there's any questions, please just come up, I believe, to this mic, correct?
Hi, I'm Sarah Burrell from REGENXBIO. First of all, thank you, everyone. This is very interesting and informative. I had a quick question. Carter, I believe you mentioned at the beginning there's over 2,400, I think you said gene therapy clinical trials.
Cell and gene clinical trials.
Cell and gene clinical trials ongoing. I guess my question is, we also know there's only a handful of approved products at this point. I'm curious, for everyone involved, like, what do you view as the reason, or perhaps, what is contributing to the kind of that large differential in terms of clinical trials and approved products at this point?
That's an interesting and certainly a somewhat scientifically complex question. So maybe I'll give the simplified answer, understanding the complexity. I think when you look at, generally speaking, we're still overall clinical trial success. If you look across all products, it's still like a 1 in 10 chance, right? And there's variability. If you're doing rare diseases, that probability goes up. If you're doing, smaller patient populations, it goes up. If you're doing cancer, it goes down. So there's lots of variables, and actually our industry analysis team actually has a great paper that looks at clinical trial success rates across all of those different, and breaks it down in a variety of ways to help us better understand. So I think when we're looking at the large number of cell and gene therapies coming through phase 2, one thing we're going to want to understand is that percentage of clinical success, how is that marrying up with historical clinical trial success rates? So you at least want it to be on par, if not better. So I think, there's going to be significant learnings about that in the coming, months, days, years. So I don't know that I have an answer now, but that's certainly something we're going to be looking for. Does that sort of answer your question? Yeah. Any other questions from the audience? Okay. Well, we've got about 3 minutes left, so I think it'd be great to do maybe just a 30-second closing argument of like what is it one thing you really want the audience to understand from your perspectives, and I'll do the same. So maybe we'll go into reverse order. Colin, what is the one thing you want this audience to really hear from you about today?
So I'd just like to say I'm here today as a voice for everyone living with a rare disease, not just the Duchenne community. The evidence is clear that patient advocacy is a crucial part for the rare disease community. My family is lucky to have Parent Project on our side, but many families dealing with other diseases don't have that luxury. They're on to advocate for themselves or their children. So I hope to be an advocate for those families and individuals in addition to the Duchenne community. And as we know, with the ongoing COVID epidemic, we all know that medical research is important to everyone, not just the rare disease community. So eventually these gene therapies can help people living with more common diseases such as Alzheimer's and Parkinson's. So in closing, I'd like to thank everyone for taking the time to hear my story, and just realize how being, including community, can be a powerful, positive influence.
Well, I, on behalf of everyone, I thank you for your participation today. You are a wonderful patient advocate, and I look forward to working with you more in the coming years. So I'm just glad I got to meet you, and I look forward to working with you. Nina.
Thank you.
Yeah, so I'll just say that adding on to what Colin said, I think key to successful collaboration are including the voice and participation of the patient and their parents, families, and caregivers and patient groups. And so engagement with patients in a meaningful way helps sponsors and all of those engaged in therapeutic development to have an understanding what matters. And so with that, thank you for including me here today to speak on behalf of both REGENXBIO and the Pathway Development Consortium. And we look forward to updating you on our work through the consortium in the coming months.
Thank you, Nina. I look forward to learning more and more about that consortium and its output. So again, look forward to working with you as well. I guess for my part, my closing arguments would be, I think clearly, as I stated in my opening remarks, this is truly an exciting and transformative time. We are in real time building precedents. This is an opportunity to build understandings that are going to chart the path forward. It's also an opportunity to think globally and build harmonized approaches to cell and gene therapy. Like again, in my entire lifetime of working in this industry for the last 15 or so years, this is the best opportunity to really build things and build them in a harmonized way, not just here in the United States, but around the world. So what does that mean for everybody in this room? It means that you should be doing exactly what you're doing now, being involved in your state organizations, being involved in your national organizations such as BIO. These are the voices that are going to be at the tables. These are the voices that are going to be carrying your thoughts forward, your insights forward, and making the change that we all want to see to ensure that we have the best and cleanest and most effective and efficient path forward for getting these medicines in the hands of the patients that need them. So stay involved, I guess, is basically my message. And with that, I believe I'm supposed to say, now it's time for lunch, so you can exit, enjoy your meal, and again, thank you for having us here today, and I hope you have a great rest of the meeting.